Cell membrane. Let's start to look at these beautiful movies. It's a very beautiful movie, isn't it? In the middle, there is a cell membrane, lipid bilayer, and downside is the intracellular space, and outside is the extracellular space. There are many proteins are floating around, and sometimes they can bind to the surface. And there are some proteins is penetrating the cell membrane and showing some the portion onto the outside, some portion inside. Now let's summarize the membrane proteins. On the cell membrane, there are some proteins facing inside, or some membrane is bound on the surface, but it's extruding out to the extracellular matrix area. Some proteins forms this kind of the penetrating protein. Some proteins are channel structures. Some proteins are transporters. And approximate thickness of this membrane is about 4 nanometer. Well, in comparison, the antibody height is about 10 nanometer. This is a good picture you want to keep in mind. Here is a summary of those channel and transporter. Channels here, this channel allows flow of those molecules. It's a very fast movement of those molecules either inward or outward. In comparison, this kind of a carrier or transporter molecules, these proteins can transport only one by one. Take one and then put this molecule inside, and the other way around may be taken here and then moving out. So these two proteins, the difference is channel is much faster and transporter or carrier is a step-by-step movement. Membrane is very thin comparing to the size of cells. That's why it can be very flexible. To provide those kind of a structural rigidity inside the cell, there are cytoskeletal proteins such as actins or tubulins. Outside of the cells, there are also many the cytoskeletal structures. So collagen is the representative ones. Also, outside of a cell membrane, there are decorated by glycans. Glycan is carbohydrate. Also, on the cell membrane, there are some small domain which is much rigid and thicker than other area. This area is called as a lipid raft. This is like a raft floating around on water surface. This lipid raft, the structure has proteins embedded. Also, cholesterol are embedded here. That's how this whole structure is thicker, also rigid. The role of the cholesterol is mimicking these stones on the wall. So if you put this kind of a stone on the wall, it makes the wall the much more rigid structure. So similar way, the cholesterol insertion into the membrane make that domain rigid. Now let's compare the ion concentration difference between in and out of the cells. If you look at the intracellular space, the sodium concentration is about 10 millimolar, but extracellular space is more than 10 times. Opposite way, potassium is much higher in intracellular space, and extracellular space is much lower. But total amount of this sodium-potassium concentration is quite similar in both sides. That implies that this sodium and potassium keeps the osmotic pressure balance. So we can imagine that originally outside of the cells, it has a high sodium concentration environment, but the cells intentionally take in the potassium into the cell and then pump out sodium to the outside, and then make the two space is quite different. Magnesium case, the difference is relatively small. So outside is a little higher, but inside is also high, and these magnesiums are usually make a complex with the DNAs. In comparison, calcium is dramatically different. Outside of the cell, the calcium concentration is reaching almost up to millimolar concentration, but inside the cell, that is less than 1 micromolar concentrations. So this more than 10 times, 10,000 times the difference make inside and outside is very uniquely different. Chloride concentration is also outside is much higher, but inside there are a lot of phosphate can make this charge balance. Now, by looking at this the data, you can think what can be the original environment of the cells in the beginning. Maybe the clue comes from the current composition of seawater. Even though the current seawater is almost 3 times saltier than extracellular matrix environment, but if you look at those relative ratio, it's surprisingly similar to each other. There may be many explanations for this, but one very attractive explanation is maybe at the time when the cells were invented, the seawater concentration was similar to the current the cell extracellular environment. That means seawater was much more diluted at the moment. But over the time, the water circulated, it goes up to the cloud, and then rain dissolve those minerals from the land, and then they accumulate more minerals into seawater. It's quite attractive, isn't it?